Hello and welcome to the C. diff Conference and Health Expo. Thanks for stopping by my poster. I'm Heidi and I lead the Translational Biology group at Rebiotix, a Ferring company. Today I'll be telling you about gut microbiota changes associated with clinical outcomes in the PUNCH CD3 clinical trial of the Investigational Live Biotherapeutic RBX 2660. RBX 2660 is a liquid suspension of a broad consortium of microbiota that has been evaluated in over 600 participants over 6 clinical trials. The trial discussed in this poster, PUNCH CD3, was a phase 3 randomized double-blinded placebo-controlled trial. I won't be presenting the final clinical outcomes of safety and efficacy, as those have been presented elsewhere by our clinical team. As detailed in the methods, treatment success in this trial was defined as absence of recurrence of C. difficile infection at 8 weeks after blinded study treatment. Participants that did not have a recurrence at 8 weeks are referred to as responders because they responded to either RBX 2660 treatment or placebo treatment. And concurrent with clinical monitoring, participants were asked to voluntarily submit stool samples through home collection, and samples were collected prior to study treatment, which is baseline, as well as 1, 4, and 8 weeks, and 3 and 6 months after receiving study treatment. Stool samples were submitted for DNA sequencing, and from that we determined microbiota composition and could look at changes over time. Sequencing data are summarized as operational taxonomic units, or OTUs, which are units used to group closely related bacteria. And this allowed us to calculate relative abundance, or proportions, of different bacteria in stool samples. And we show this data at the taxonomic class level. We also calculated alpha diversity, which is a measure of the number of different bacteria within a sample. And in general, restored gut microbiomes have a higher alpha diversity. And we summarized our microbiome analysis and findings using a prototype biomarker that we developed and called the Microbiome Health Index, or MHI. The RBX 2660 clinical efficacy graph summarizes the clinical outcomes from the PUNCH CD3 trial, and the key takeaway is that RBX 2660 was found to be superior to placebo. We had a predefined definition for success, and we met that. And these are really incredibly exciting results, not only for the field, but also in evaluating how investigational product behaves in the clinic. And building off of this excitement, I'd like to share with you some of our microbiome analyses. So in the first set of graphs, we're summarizing key microbiota changes by looking at the class level of several bacteria known to be associated with either health, wellness, and absence of disease, or conversely, associated with poor health outcomes. So along the y-axis, you have relative abundance, and along the x-axis, or horizontal axis, you have the time course of the study. The last sample in each of these is the RBX 2660 product, which represents the restored microbiome. And so if you look at baseline, what you see in RBX 2660 treated responders is that the baseline of Bacteroideia and Clostridia are both very low, whereas Gamma proteobacteria and bacilli are shown in overabundance. And as you move across the time course, we see a shift in the microbiome composition in the RBX 2660 treated responders, where the microbiome composition is starting to look more like that of RBX 2660. We see Gamma proteobacteria decrease as well as bacilli decrease. If you compare this to the placebo-treated responders, there is an increase in Bacteroideia, but it's not as extensive or happens as quickly as in the RBX-treated responders. Gamma proteobacteria and bacilli both decrease in the placebo-treated responders, but again, not to the same extent or as quickly as in active treated responders. So the RBX 2660 treated responders demonstrated a more rapid and more extensive recovery of Bacteroideia and a decrease in Gamma proteobacteria. And again, this was relative to the placebo-treated responders. And these findings are important because bacteria in the Bacteroideia and Clostridia classes are known to be associated with health and wellness and absence of disease. Conversely, bacteria in the Gamma proteobacteria and bacilli are known to be associated with presence of disease, and many of them commonly harbor antimicrobial resistance genes. If you move to the second set of graphs in the microbiome analysis, we really try to drill down into these results with the longitudinal analysis. And there were some key differences between RBX 2660 and placebo-treated responders. And this is really the clearest way to show active versus placebo responders because we wanted rigorous statistics over a time course. And these graphs show average changes using repeated measures over the time course on the x-axis. The error bars represent the 95% confidence intervals calculated from the relative abundance, which is shown on the y-axis. And the solid symbols represent RBX 2660 responders, and the other symbols represent placebo responders. And the differences were statistically significant. If you look at the relative abundance at 0, or baseline on the x-axis, you see that RBX 2660 responders have restoration that is more extensive and rapid, and this happens as soon as 7 days in all classes. Again, in the placebo responders, we do not see Bacteroideia recover out to 6 months. Clostridia recovers, but not as quickly or to the same extent as in the active treated participants. Both gamma proteobacteria and bacilli decrease more rapidly in the RBX 2660 responders. The next graph shows the Microbiome Health Index, or MHI, which is a biomarker that combines the 4 key taxa in the previous graphs into a single metric. A higher MHI score is associated with wellness and absence of disease, while a lower score is associated with presence of disease and poorer health outcomes. The dotted line across the graphs represents a threshold above which predicts a more restored microbiome with better health outcomes, and below the dot predicts a more dysbiotic gut microbiome and poorer health outcomes. And note that the data are presented on a log scale on the x-axis, and this is over a time course on the y-axis, starting at baseline, which is before RBX 2660 administration through 6 months. At baseline, participants had a low MHI score, especially when compared to the MHI score of RBX 2660. And as early as 7 days, the majority of participants that responded clinically, which shifted above the dotted line, that threshold that predicts a healthier gut microbiome composition. And these changes persisted out to 6 months. So in general, RBX 2660 treated responders trended higher than placebo-treated responders. Below that is the alpha diversity, and this was statistically significantly increased in all post-treatment participant samples. So in conclusion, in addition to the clinical superiority reported for RBX 2660 over placebo, in this trial we observed significant microbiota changes from before to after treatment toward healthier microbiome compositions, and these changes were more extensive in RBX 2660 responders than in placebo responders. Thank you for your time.